Okay, can we start then?
Okay.
Okay, hi everyone. My name is Camila. I'm a senior TA here at PPCR. I was a 2014 PPCR study, and since I finished the course, I started being TA, so it's a pleasure to be here doing this optional lecture. Thank you for coming, and as Sueli says, good morning, good night. We don't know where you are all over the world. But I hope you enjoyed the lecture today, as we all enjoyed the lecture yesterday from Professor Fragni. So today we're gonna review and discuss together two topics. One of them is adaptive designs. And in adaptive designs, we're gonna discuss interim analysis, and we're also gonna discuss N-of-1, okay? So I have no financial or commercial interest involved in this presentation, and we can go ahead and start with the lecture outline. So we had for this week two case studies. One case study talking about insomnia and selenium, and another case study talking about fibromyalgia with Dr. Sato and the idea of doing or not a N-of-1 type of study. And we're going to discuss a little bit of clinical evidence and where these studies fell off when we talk about medicine-based evidence, okay? So to start, I would like to see if some study wants to quick summarize for us the first case study. That was the case of the Selenium trial, and if they decide to plan or not an interim analysis. Keiko, do you think one study can summarize the case for us?
Maybe Jason.
Yes, yeah, thanks.
Sure.
This is, I forgot the name, but a guy from the pharmaceutical company is designing a RCT which investigates the selenium, which is a sleeping pill, and it is going to run for 24 weeks, for 4 weeks to evaluate the effectiveness and safety. And after all the discussion, the advisor from the university, which was Professor B, I think, thought that they forgot to mention about the interim analysis, which the professor thought is important, and they are going to have a further discussion about whether or not to have the interim analysis, and if yes, then which method to adopt.
Mm-hmm. Which method to use for the interim analysis, correct? Yeah. As we are talking already and you summarize the case, can you define for us what do you understand about what is interim analysis?
Interim analysis is like we have the final analysis, which is our goal in a study, and the interim analysis is any analysis between the start of the study and the end of the final analysis, which is done by a independent group and with full access to the data. Mm-hmm. But which might, and with those analysis, the investigators might determine whether to continue or stop the study, and also to assess the dropout rate and any other concerns of the study.
Yes, exactly. That's a very good explanation. So interim analysis is an analysis, exactly as you said, that is done during data collection, and is usually part of this trial where you plan the interim analysis we call adaptive design trial, and this would be a group sequential type of trial in which you plan interim analysis and you also plan decisions to take about the trial course based on the results of that interim analysis. So every time that you're thinking about doing an interim analysis in your trial, you also have to think about why you're going to do, and of course all the methodology, but the discussion and the decisions that you're going to make in the clinical trial after you have the results of the interim analysis. That was perfect, Jake. And I don't know if you want to continue or if Keiko wants to appoint another student. But now let's think about why do we want to do an interim analysis. You kind of talk a little bit about it, saying that we do analysis to see if we stop the study, if we assess sample size. But in terms of a clinical trial, why this is beneficial to do an interim analysis and not do only an RCT as simple as we saw on module 1, right? Using a parallel design and just doing the final analysis and so on. What would be one of the reasons people decide to do interim analysis in clinical trials?
Okay, Jason, if you like, you can continue, maybe.
Yeah, thank you.
Yeah.
And I think from the book, we can see 3 major reasons. Those are efficacy, safety, and futility, which we might want to evaluate the intervention over the control and to see whether we need to continue or stop the study. And besides those, we also, the aims of the interim analysis also contains the monitoring of whether the study is on the designed track, like whether the dropout rate is very, very high, and also we need to investigate and intervene into those factors which affect those stuffs.
Yes, that's a very good explanation, right? So we can do interim analysis usually for efficacy, safety, or futility, but and we know that it's important to do interim analysis, as you point out. You pointed some advantages of interim analysis, but why do you think clinical trials use interim analysis? Because there are also disadvantages. Why, in this case of the Serenium, why they think they should do an interim analysis? They think about they should do just because of safety, or for instance, they think it could be an ethical problem, or they are worried about how much the clinical trial will cost also, besides knowing the advantages or the disadvantages. What do you think about that?
In this specific case of our case study, I think one concern from Professor B is that if it is very effective, then it is of ethical concern not to give the control arm the drug which is investigated. Yeah.
So in this specific one, they were concerned about the ethical issues, right? They would say they are studying insomnia. So the maximum number receiving the best possible treatment at the early stages, it's better, right? Yesterday, Professor Fragni discussed some cancer trials with us. So this type of interim analysis is common in cancer studies, because in cancer studies, we all know that there's new treatments coming all the time. There's no specific treatment, and for different cancers, the treatment can be different. So if you can get a new treatment on the market available for everyone as fast as possible, it's better, right? So in this case, interim analysis are considering for these ethical issues. Agree? And also, they discuss in our case study, and it's frequently discussed in interim analysis cases, costs. Because pharmaceutical companies, clinical trials are very expensive. If you finish the trial earlier, you can save some money, right? It's for us that do most of our trials are, at least for me, most of the trials that I participate are investigator sponsor. Even though we worry about the cost, making money at the end is not our main concern, right? But we know that pharmaceutical companies and drugs that we buy, they all have behind political and cost reasons to be there. So many trials and many companies consider this when they plan an interim analysis. Does anyone wants to discuss something a little bit more the case, or can we move on? I know there's more students here, so if you have any doubts now, we can talk a little bit and discuss it.
Okay, maybe I can find one question from Julia. Julia, can you talk? Can you talk? because I put some answer, but maybe you should ask your doubt to Camila directly, if you like.
Yeah, hi Camila, this is Julia
Hi.
from Brazil. Are you listening me well?
Yes, I can hear you, Julia.
Okay, so yesterday my question is if it is possible to do an interim analysis without considering the trial an adaptive trial, and because although like I think the purpose of doing an interim analysis is to adapt the trial, but the purpose of my question is because yesterday during live lecture I saw someone, I think it was a TA that wrote that it was possible to do an interim analysis in RCTs, for example. Then I lost all the track of my reasoning, you know.
But then what was the reason why you're doing the interim analysis?
Oh, I don't know. I just asked the question because I lost I didn't get it, the purpose. Then just to run in an RCT, you know, since you're not going to change.
Exactly, but then you have to think, you're doing analysis that have some advantages, of course, but it has some disadvantage, right? So if you're doing analysis just to see if your data is going good, so what that means, understand?
Yeah, of course.
You would have to have even though you could say, I'm going to do an interim analysis to see if my study is on track, and I'm not going to change the study regarding my interim analysis. You plan that.
Yeah, that is risky.
That is risky because you remember, you're going to have to plan your interim analysis, you're going to have to plan your end of study analysis, and you're going to lose some things, lose meaning like you might have conflicting results in the middle of the trial. And if you did not plan what you're going to do with these conflicting results, it can backfire on you, right?
Yes.
So yeah, when you do an interim analysis, you are doing an adaptive design. Even if you plan that your interim analysis will not change much your trial design later on, when you have a result, you have to deal with the results. Imagine like you plan an interim analysis in the example of the John Luke, and you were wrong, and actually now you're in your interim analysis, you saw that placebo is much better than Serenion, and people are getting side effects related to suicidal ideation, for instance, right? And you just plan to do the interim analysis. What you're gonna do for trial, understand? Like it's not safe anymore to do it, or so on. Exactly. So it's tricky because when we think about interim analysis, we don't think about adaptative designs, right? But it's a type of adaptative design, yes.
Yeah,
because
totally agree.
Adaptative design, we're going to discuss a little bit later, but adaptative design means that doing analysis in the middle, review that analysis and take a decision, and then continue the trial or not, and the decision could be stopping the trial, yes.
Got it. Thank you. And Camila, I have one more question. Sorry to interrupt for the class. Sorry, I don't know if you're going to discuss it later on, but my question is, I didn't understand the point when we're accessing and we're doing interim analysis by safety, there's no penalty to p-value, but when doing it for futility and efficacy, there is.
Sorry, can you say again, when doing for
safety issues?
So usually when you do an interim analysis for safety issues, safety might not be your primary outcome, right? Your primary outcome is if the drug is better than the control, but you are worried about safety. So you're not assessing your primary outcome. In this interim analysis, you're not going to compare efficacy. Understand? You're not going to look to that data. You're going to look only to the safety data.
Ah, okay. So the purpose of only the final analysis is to look at efficacy. Otherwise, you're going to analyzing the data, and that is why you have the penalty. Okay, got it. Okay.
Exactly.
Oh, thank you.
Yes, but for instance, if a study has as main outcome safety, this is not common, okay? Let's just think about, invent an example just to think together.
Okay.
if a study has as main outcome safety, meaning in my N-of-1 and in my final analysis, I want to show that the drug is safe, and I do an interim analysis for safety, and I'm going to look to that same outcome, I'm also have to use an alpha spending function, because I'm looking at that outcome 2 times.
Okay, yes, got it.
Yeah. Perfect.
Okay, thank you so much, Camila.
Yes, that was very interesting questions, actually. Yes.
Thank you.
Does anyone wants to make more questions now, or can we move on and we'll make more questions later? Okay, so we'll consider the silence that we're gonna keep going, okay? So we already talked about the options that we get in this case study, so I'm gonna move on. So we have some options that was to do nothing, no interim analysis, do an interim analysis for safety only, so not looking at efficacy, therefore not paying the p-value penalty. We just discussed this with Julia, and doing an interim analysis for safety and efficacy, right? And then we would here in the case study, we had the possibility of doing 2 different alpha spending functions. Then we're going to discuss a little bit later more about this alpha spending functions. Okay. As Jason, I think I'm pronouncing your name correctly, summarized for us. The main types of interim analysis are usually for safety, efficacy, or futility. Okay, they are different interim analysis. They require different types of planning. And we already discussed a little bit about the advantages and disadvantages of doing interim analysis. As always, one thing that we learn in PPCR is that there is no 100% right and 100% wrong in everything that we do. When we have a clinical trial, we gain something, but we also have disadvantage. So we need to think about both of them and put them on a balance and see what's better for your research question, right? It's not what's better for clinical trials in general, but what's better for your research question. So talk a little bit about the advantage, as we discussed about the cost of the trials and the risk. So an interim analysis can shorten the duration of a trial, can show if the treatment is safe before we finish the total sample collection, right? Can tell us if that drug is efficacy or not efficacy, like meaning futility, and decide if we continue or stop the trial, and can give us a better effect signal. As we discussed yesterday, some interim analyses are designed to reassess the sample size of the trial. So you are sure that when you calculate your effect size, meaning your mean difference, the expected mean difference and expected variance of a population, you did a good job. This is common sometimes in studies when drugs are made in a specific population, and now you want to test the drug in a different population. For example, it's very common to have HIV studies on Africa, because the amount of population that are HIV positive there, it's bigger, and the concentration is bigger compared to other places of the world. But then when you get that drug and you come to another country that the population might be a little bit different, for instance, if you go to China, you might want to reassess the effect signal, because the population is very different. Even though you did a big trial, the standard deviation of your population, the variation might be different, and it would be a good idea to reassess your sample size in the middle of the trial to know if you have power to finish that. Okay. Some disadvantages, as we discussed already too. We have statistical versus clinical significance. Of course, we do want to know if the drug works and put on the market as fast as possible, as we are seeing now, right, with the COVID vaccine and some COVID testing that we discussed yesterday in the lecture. We also can have increased chances of type 1 and type 2 errors, because you did not have your full sample size yet, and you can have false positive rates too because of that. You can lose some power, of course, for type 2 error, and sometimes it's a challenge for the scientific community to accept. It's very common that people can look or be more critical about a result if the trial finish because of interim analysis. But again, there is no right or wrong here. The importance is if you plan to do an interim analysis, it has to be well planned, okay? One doubt that I usually have, and it's not that straightforward to explain, but I will try to explain a little bit here, is why we need to use the alpha spending, or why we need to change the p-values and reassess the data, okay? When I try to understand this, I usually bring the examples in my head of, for instance, when we have to do subgroup analysis, for instance, and we do Bonferroni correction. So we correct the p-value, we divide the p-value for the amount of analysis that we're gonna do. When we think about interim analysis, I like to think a little bit like that, right? So we need to do some changes in the p-value because we are analyzing the data more than one time, right? And we all know that even though it seems a little bit arbitrary, considering a p-value of less than 0.005, it's well accepted in the literature, and it's well accepted because you are saying to your readers that you have a chance less than 5% of having a false positive result, meaning finding statistical significant result that is not there, right? So when you're analyzing your data, we usually consider an alpha of right, a p-value, an alpha of 0.005. So the first time that you look your data, you have a probability of having an error of 0.05 from 100%. Sorry, it came back my slide. The second time that you look your data, you already spend your first 0.05. So now you don't have 100% anymore, you have 0.95. And if you calculate 5% of like your new probability here, you're gonna have a probability of 0.0475 to have to commit an error. Meaning, if you sum the total probability of the first look, that was 0.05, and the second look, now the probability of you having a false positive result, it's higher. It's almost 10% here. So you double the probability of having a false positive result because you look to your data twice. Same thinking that we think about when we do Bonferroni, right? When you do two comparisons in your data, you divide your p-value by 2. Here you increase your chance of error by almost 2. That's why alpha spending functions are used in interim analysis to be sure that when you look to the data more than one time, you are considering that you have more chances now to find a positive result when this result does not exist. Okay, I hope this is clear for you guys.
Can you please explain what happens if you see the data twice, if you do interim analysis twice to the type 1 error? I understand now it has become 0.0475, but what happens if you do another time?
So then you're gonna have to do the calculation one more time, right? If you think 0.47, now we have 0.95 minus 0.047, and let's make this calculation very quick here. 1 second.
Can you go back one slide and explain?
Yes, of course, I can go back one slide. So this is one example, right? The rules, they deal, the alpha spending rules deal the p-value differently, but this is the probability of you looking a second time, right? So now you spend 0.047. So we go 0.095 minus 0.047, we are gonna have 0.091, right? 91. Now the third time they're gonna look to the data, you're gonna have 0.91 times 0.05, correct? Because now is your new probability. 0.91 times, let's do here, 0.05. Now we're gonna have a probability of 0.048. And if you sum all of them, you always have to sum, right? It's gonna be 0.0048 plus 0.047 plus 0.05. So now we're gonna have a probability of having, one second, I did the calculation wrong. 0.048 plus 0.047 plus 0.05. Now our probability here is 0.1. So it's, you see that it's not an exponential increasing. Actually, it's not a linear increasing. It's like more an exponential increasing in the meaning that now instead of having 9% of chance, you're gonna have about 10% of chance to find a positive result when that result does not exist. So it starts increasing slowly because your number here, right? Your number here will decrease because you are losing that alpha spending. But every time you look to the data, you're gonna increase the chance of finding a result, a positive result that does not exist by an amount of percentage. And for the first to the second, you almost double the chance, and later on the chance is gonna get smaller, but you're still gonna have it. It makes sense. Ahmed?
Yeah.
Yeah. So every time you're gonna have to calculate again to see how much your rate of making a mistake it's increasing for every time you look to the data. This is just a probability example. That's why the alpha spending functions, they take in consideration using a lower p-value in the beginning. For instance, they don't do this calculation right away. This is most for us to understand that the probability changes every time you look to the data. The probability of you finding a result, a positive result that does not exist, changes every time you look to the data. That's why you use one of the alpha spending functions, because you want that probability to be as minimal as possible in the beginning. So when you get to the end of the trial, you still have a reasonable p-value to achieve. Does it make sense?
Yes, thank you.
Yes, are you sure? Okay. So for alpha spending functions, we have different methods, right? Don't worry too much about these methods, like how to calculate and how to do things. Try just to understand the reasoning behind these methods, right? I think that's enough for now, and we can talk more about a bit later. But there are three main methods that uses. That is the Pocock method, the O'Brien-Fleming method, and the Haber-Perot method, right? And for your case study, if I'm not confusing, they were considering the Pocock and the O'Brien-Fleming method. Does anyone wants to jump in and tell us what they understand about these methods and if they want to summarize the differences? Keiko, do you think we have someone?
Yeah, please try from China, like Olola. Can you talk?
Yeah, yes, I can talk.
Please.
Yes. Do you want just to summarize the methods? You don't have to tell exactly the p-value, but just what do you understand by this and what do you understand about some advantages and disadvantages of some of them. You don't have to talk all of them, just.
I think the Pocock approach is a fixed nominal level approach, and it sets the same value for each interim analysis. And the second one is it's widely adopted because the stopping criteria are more conservative at the earlier stage. And the third one in this approach, the trial is stopped when there is overwhelming evidence to stop the trial, and the threshold has been considered at 0.001.
Mm-hmm. Great explanation. Thank you so much. So you summarize everything. There's nothing left for me to say. Thank you. So exactly like that. So all the methods have different ways to deal with the alpha function, and all the methods have some advantages and some disadvantages. As you explain how the methods work, we can talk about the advantage and disadvantage together. And I just put it here exactly what you say in this graphic, right? The POHOC keeps a stable p-value. The payroll method, a lower p-value in the beginning, but at the end it comes back to the normal one. And the O'Brien framing has a more conservative approach, making much lower p-value in the first analysis, and this p-value starts getting more losing out as the analysis keep going on, right? Do you want to talk about some disadvantages or of any of these methods, or for instance, if they would be used more in which type of trial and so on?
Anyone? Aurora, can you continue?
Oh,
No?
sorry, my internet is not stable. Can you repeat the question?
You are doing such a good job. You summarize all of them for us, right? Now, can you think about some advantages and disadvantages of these methods?
I think the for the O'Brien Fleming approach, and it's the border of this approach is large, and and for the second, and for the Pocock approach, I think it's a fixed nominal level, so maybe it's easier to use. I don't know, but it's difficult to report or to report the result.
No, that's perfect. Thank you. Yeah, as you said, like the Pocock, it's easier to interpret and use because you have the same p-value along the trial. So you divide your final p-value by the number of analyses that you would do. It's in some disadvantages, like you increase the chance to stop the trial earlier, right? Because if you see here for the first analysis is the one that has the most loosened p-value. So you have to be careful when you use, because if you use for efficacy, for instance, you have higher chance to stop the trial early for efficacy and might not really have your real results by them. But it's a method that are commonly used for futility, for example, right? So the O'Brien method is one method that is mostly used for efficacy here. The chance of stopping the trial early, you have a much more conservative p-value. So it's a method that it would be much more accepted by the literature if you end up stopping your trial earlier because of interim analysis. But again, it has a disadvantage. The same a way that having a much lower p-value in the beginning is an advantage, it is a disadvantage, right? Because with a much lower p-value here in the beginning, it's much harder for you to achieve this p-value unless you have treatment that is much more efficacious. And the payroll type of approach, it's common in some device studies and cardiology studies. Actually, it's a kind of a common approach. It has a little bit more conservative p-value in the beginning when compared to the Pocock, but also you don't you don't lose too much in your final analysis. Okay, is there any questions here about these methods or where they are used?
Well, I think we're okay. Anyone? Maybe we can go ahead.
Okay.
And then if I can find it, the question, maybe I will call you.
Yes, okay, thank you, Keiko, yeah. So for this trial, I know you replied the poll yesterday and during during the lecture, right? But what would you do? Does someone wants to tell me what they chose for this study? Maybe Keiko, you can come with someone that can tell us
Yes,
what they chose and why.
Yes, Gong Shari from China.
Hi.
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 If I am professor, I will choose three point. Because I think do an terminal analysis may be increase the successful of clinical and make the study more efficiency. And when we do a trial that is not efficacy or safety or futility, and then I can be early to stop my trial.
Mm-hmm. Okay, so would you choose to do then do nothing?
No, three.
Okay, sorry, cut it at the beginning. Okay, so you would do safety and efficacy. Okay, that's very nice. Do you think of which one of the alpha spending functions would you choose?
Pardon? Sorry.
If you have to choose one of the, let's imagine that you chose to do this study of the Selenium and you decided to do the interim analysis for efficacy. Which one of these alpha functions would you choose? There's no right answer. I just want to know your opinion.
一个 O'Brien Fleming.
O'Brien Fleming. Yes, that's a nice approach. So you would choose a more conservative approach, meaning that you would stop your trial only if you have a bigger difference between your active treatment and your control treatment. That's very good. Thank you for sharing with us.
Oh, thank you.
Yes.
I have a question. I know we discussed about these three methods. I also read about Armitage McPherson method, Rove method, and Bayesian.
Bayesian approach.
Yeah, I was trying to understand, but I was not able to put together.
Yeah, so I did not bring a slide with different methods because these are the most common ones, but there are more methods, and they all based on the same. You have just to remember that they all based on the same thing. That's in the probability of you finding an error or in the probability of you finding a result that is not there when you look to your data too many times. I'm not so familiar with the Hermitage. I'm not so familiar with these two methods, but I can prepare something and send by email later on. But I don't have here to explain right now, and by heart, I don't know what exactly what they mean.
A lot of articles we were reading were using Bayesian model, so I don't know.
Okay, yeah, I'm just seeing if I find something here. But to be honest for you, for now, I don't know how to explain this for you, but I can look into it and send later on. But a Bayesian type of analysis is also a probabilistic analysis, right? So we'll probably use the Bayesian method to calculate the probability of you finding a result that is not there, a positive result when you don't have that result, using the Bayesian method. That is a method that is used for several other things, not just for calculating alpha spending functions, okay? But I can go ahead and look more with calm, and if we have time, we can come in the end. But it will be that they will use like the Bayesian theorem to calculate the alpha spending function. Okay, I know this does not reply your question, but that's what I have. Is that okay?
Yeah.
Yeah.
Thank you.
You're welcome, yes. Okay, so let's move on a little bit. Is there any other questions so we can move on? If there is, just tell Keiko and Keiko will tell me, okay?
Yeah, just if we have some time, maybe an notice question, like superiority, inferiority, and the equivalence trials. Maybe you can ask. Can you?
Yeah. So, so which methods would you choose if you were to choose superiority? Would you choose this thing, Peto method, or in order to show like Haberl-Peto method would be good for superiority trials? I just want to know, how would you choose the method based on superiority, inferiority, or equivalence, or is it that has nothing to do with this?
So yeah, here we are thinking about two different concepts, right? Can you do an interim analysis in a superiority trial? Yes, you can do an interim analysis in a superiority trial. And the method, it always depends on, again, the method always depends on your research question, right? But usually when you are trying to do a interim analysis for efficacy in a superiority trial, I personally would choose a method, and it's very common in the literature, using the more conservative methods, going for the blind framing or the Haberl-Peto method that are more conservative, at least in the beginning of the analysis, right? Now, if you're doing a non-inferiority trial, meaning you are calculating a non-inferiority margin, and you're doing an interim analysis in your non-inferiority trial, if that's what you mean?
Yeah.
Yeah, because you understand there are different things, right? One is your trial design. There is a non-inferiority trial, and it's a non-inferiority trial with an interim analysis. So to be honest, I don't think that's very, very common, because besides of defining a non-inferiority margin, you're gonna have to define the p-value to evaluate the non-inferiority margin more than one time in your trial, and this can get really complicated, right? But for this, I also would use the most conservative methods, right? You see the POCOC method more in trials that are assessing safety, for instance, because you don't want it to be too rigorous with safety and not finding that your drug is not safe in the beginning because you are too rigorous with the p-value. The other two methods are more used for when you are assessing efficacy or futility. Okay.
Okay. So from what I get is there is definitive answer for superiority to use a conservative method, but using interim analysis in non-inferiority is kind of too rare, and that's what I got.
It's kind of too rare, right? Because now you are testing two things. Besides of testing non-inferiority, you are adding another layer in the non-inferiority. So you're gonna have to add the layer of the p-value in the non-inferiority trial. Like it's not that common, to be 100% honest with you. Yeah, non-inferiority trials by itself are not as much common as superiority trials. Nowadays, they're getting more common, right? But adding on top of that, an interim analysis, it wouldn't be common at all. And I think if they do, they would follow the same idea, having a more conservative approach. Usually the less conservative approach is used more to safety type of trials. Okay?
Okay. So, so is there any reason why we would use POCOC method at all?
Yeah, so for safety trials,
for safety trials.
It's very commonly used for safety trials. I'm not saying it's not used at all for efficacy or futility, but there are a lot of discussion and how can I say, it's not a method well seen when used for efficacy trial. Okay, people will maybe be not so comfortable believing in the results of your trial if you use a POCOC method for efficacy, because the p-value in the beginning is too loose. So the chances of having a false positive results are way higher here, right? So it's used mostly for safety.
So more of phase 1 trials then.
So yeah, we think about phase 1 trials being safety trials, but that's not really that, right? You can also assess safety in phase 2 trials. It's not common at all doing interim analysis in phase 1 trials. This is something that you do most on phase 2 and phase 3 trials. So you have an idea of safety. You know your drug, it's ideally safe in humans, right? But now you are expanding your population. And you want to still assess if that drug is gonna, in this expanded population, have the same rates of side effects or more side effects than you saw on your phase 1 trials. Usually in phase 1 trials, you do not do an interim analysis. It's a much smaller trial. Most of the times the first contact of that drug or that compound or procedure in humans, right? And most of phase 1 trials, you assess your drug in healthy patients, not in your target population. Some phase 1 trials do that. So it would be more common in phase 2 trials that assess. Okay. Yeah. Okay. Okay. Let's check time. Yes. So this is, as we already discussed, an interim analysis is a method that is used in an adaptive design trial, right? And what are adaptive designs? So a traditional fixed sample design trial, we would have the design, we would make the trial and make the analysis at the end. And in adaptive design, we have the design, we conduct the trial, and while we are conducting the trial, we review the trial, adapt, and continue the trial, and also do the final analysis, okay? So there are some advantages here. There's a flexible trial, like you break some conventions, but there are a very disadvantage in a way that we need to think about it is like the need to preserve statistical rigor and trial integrity when you're doing adaptive design. It's important for us to remember that everything needs to be well planned. You guys are seeing the group project now. All trials are planned in details, right? We need to think about the limitations of our design and how we're going to address those limitations. And if something happens in our trial, how we're going to address what happens. And it's the same thing for adaptive design. It doesn't mean that you can run a trial and change in the middle, right? It means that you need to plan your design, plan how you're going to change, plan how you're going to analyze our data and what you do with that results, okay? So there are some regulations about use and guidance of adaptive designs, right? And they have like a predefined adaptive charter, like that you have like a checklist of things that you have to think about it when you're planning adaptive design, okay? So there are several types of adaptive designs. We talked some of them yesterday during the lecture. I brought here as example all these designs and the general idea, right? We're not going to talk about all of them, but just for you to see that it's not only group sequential designs in which we do an interim analysis and decide include the options to stop the trial early for safety, futility, or efficacy, but there are different types of design. For instance, the first one, continue reassessment methods, that you do a dose escalation, and you want to try the tolerance of the drug. So you can do a design in which we start the trial with one dose, and then after a while you're going to increase that dose, and after a while we're going to increase that dose, and so on, until you see the maximum dose that you can use in that trial. So trials that usually do that in the same patient, right? It's not meaning that you are randomizing patients to different doses. You are escalating the dose in that patient. Is a type of adaptive design. We also discussed yesterday the example number 3 here, the sample size estimation. And we discussed today with the example of HIV trials, for instance, where you can adjust the sample size to ensure your power at the end, right? We all did the sample size method here, and I hope you're all thinking about the sample size on your group project. We know that calculate sample size, it's not easy at all, right? Even though we are discussing previous studies, sometimes it's very hard to estimate, especially the variance that you expect in your population. So this is a type of style that could be common, right? So they are multi-arms or multi-stage trials, example of trials in which you assess the patient while he's doing the trial, and you can drop people that are not showing the results that you expect and keep people that are showing good results to the drug. This is a type of trial that's used a lot in cancer studies. So you're testing a new drug for cancer trials, and you see that some people are responding very well to your drug, and some people are responding very bad, having side effects, or not entering remission, and so on. So you would drop the losers, taking out the people that are not responding to your drug, and keep only the ones that are having good effects of that drug. Of course, there are advantages and disadvantages here. One big advantage for the patient itself, if they're not responding to the drug, you give the opportunity of the patient to look for alternative treatment and not being kept in your clinical trial. But later on, you would have to see why your drug works only on some individuals and does not work in other individuals, and so on. We have these examples here. You can go to the literature. I'm pretty sure as the alpha spending methods, we also have different types of adaptive designs, and these methods can be growing as time passes, because we see the need of having different designs, okay? So now I think if we don't have any questions, I can move on with one example. Do we have any questions?
I think we can go ahead.
Mm-hmm.
Maybe.
Okay.
Yeah.
So I know Dr. Frank gave some nice examples yesterday in the lecture, in the live lecture. I brought a different example here just for us to follow together. This is what I published in the Lancet journal. It's a journal that has a high impact factor, and it was checking this drug, ivabrandine, and outcomes in chronic heart failure. It calls the SHIFT trial, right? It was a randomized placebo-controlled study. Here is from 2010, right? And in these trials, what they wanted to check is that patients with symptomatic systolic heart failure or heart failure with reduced ejection fraction, right? They wanted to see if this sinus node inhibitor, that is ivabrandine, results in proof of heart failure related mortality or hospitalization compared to placebo. So it's a drug that can keep people that have heart problems, can reduce the chance of people with heart problems to die or have complications related to the heart. So it would be ethical if this drug is really has efficacy to be available to more people as fast as possible. Agree? And in this type of studies, you recruit a lot of patients. I think here was about 10,000 patients, if I'm not wrong. So they plan an interim analysis for efficacy, okay? For the first interim analysis in the primary endpoint, in which they evaluate cardiovascular death or hospitalization because of heart failure complications, they saw a hazard ratio of 0.77, so meaning a 23% chance, less chance of having an event in the group treatment, and a very low p-value, C0.0014. But this p-value was very on top of the p-value that they would expect for that interim analysis. Here they used the Hito method, so they were expecting a p-value lower than 0.001, okay? But this was a promising result. And when they had this result, now they would have to take a decision, right? Based on what they plan. So we can take several decisions thinking about an interim analysis, and all these decisions need to be plain, right? You could have amazing results and then stop the trial for efficacy. You can have favorable results and then decide to continue to your original sample size, right? You could have hopeful results or not brilliant results and have planned and decide to extend your number of subjects. You see that the results were not there probably because you're not power enough to show those results. Or you can have also disappointing and hopeless results. And for that, you could decide to stop for futility or harm or continue to your original sample size. Again, all of this has to be planned before. So let's see what they decide in the shift trial, and let's talk about who makes this decision. It's important for us to know too that when you do an interim analysis, even though you have all the design plan, the decision is not, it might not be so black and white, and it's not a decision of the PI, okay? The decision is from the data monitor committee or the data safety monitoring board. So you would have a board with clinicians and not clinicians that are not involved in your trial. So they don't have personal relationship to that trial to make the decision, right? So statisticians and clinicians that don't have personal decision. So there are stopping rules, and they are guidelines for the DSNB. The DSNB can decide not to stop a trial even though you achieve a p-value that is in your stopping rules, okay? In the case of the shifted trial, for instance, they got very close to what they wanted to get. The DSNB took the data and took a decision based on those results, okay? I just want to be clear here that even though they did all the design and they have the rules have to be assessed by a board, and the board will take the decision with what to do with the trial. And in this case, the board recommended them not to stop the trial, okay? And they went until the final of the study, and as you see here, the study was again favoring ivabradine, and the hazard ratio was 0.82, meaning about 18% less chance of having an event if you are in the ivabradine group. It's a very good percentage, right? If you have heart disease or heart failure, and I tell you, you take this drug and you have about 20% less chance to have a cardiovascular death or a cardiovascular hospitalization event, it's a good chance. And the p-value was very significant. They could have think about stopping here, but they did not stop. And for this trial, there was a specific reason that the DSNB decided not to stop. While they were doing the trial, another trial came up. This was a subgroup analysis of the Beautiful trial, right? In which they are evaluating the same drug in a slightly different population, but the subgroup analysis was focused in the same population. And the Beautiful trial found no impact of the ivabradine in the outcomes of these patients. So the DSNB also takes in consideration the literature that is going on and other trials that is going on along the trial that you are doing. So the board needs to take all of this in consideration. That's why the stopping rules are more like guidelines and not only rules, because it can be that while you're conducting your trial, even though you're seeing promising results, other trial, other group are showing that the same drug or a similar compound is having negative results or producing some harmful side effects. So this is one example of the SHIFT trial. And I know Professor Fragni brought other examples yesterday, but I wanted to bring this example to see that, again, it's not a so black and white decision. It's not so easy to decide to stop a trial, even though you plan an interim analysis, okay? So now in the cardiology guidelines, ivermectin should be considered to reduce the risk of heart failure hospitalization in patients with sinus infection, right? And the effect size here, it's bigger, other trials were made, and it could be considered to reduce the risk of heart failure. So now it's an accepted medication that they use, okay? Before we moving, now we're gonna move from interim analysis and adaptive designs to the case study 2 and talk a little bit about N-of-1 trials. Do you have any questions?
I don't think so.
Okay.
Maybe we can, is it okay for everyone? Maybe good for you, good for now. I think it's okay.
So Keiko, as you are here, could you point a study to summarize the case study 2 for us?
Yes. Hi, Tian. You didn't talk today, right, Tian? Can you open your mic? Okay.
And, so I wrote a very brief summary for this case. Is Dr. Sato wanted to assess optimal treatments for fibromyalgia, and he ran a randomized double-blind multi-crossover N-of-1 trial to compare anti-tryptylline and the combination anti-tryptylline and the fluoxetine in patients with fibromyalgia. I'm sorry, I have trouble with reading long words. And I did summarize the advantages and disadvantages of N-of-1 trials, but the summarize for this case is just
go ahead. Can you tell us about the advantages and disadvantages of N-of-1 trials?
Okay, so the advantages of N-of-1 trials is first, it can use clinical indicators closely related to patients, such as case fatality rate and quality of life, etc., and as the main outcome indicators rather than pure biological indicators to prove therapeutic measures that are effective for individual patients. And second, it narrows the distance between research and practice, increases the patient's awareness of their own situation, and increases patient participation in disease management. Patients and doctors discuss and decide future treatments which are easily accepted by patients and lost to, and it will reduce follow-up lost. And this method also has great potential in improving the quality of medical care and guiding patients with chronic diseases to use expensive and potentially harmful drugs wisely. And the final one is the research results obtained using this design are also the highest level of evidence. But of course, it comes with some cost. So natural changes in this condition, changes in the environment and climate and psychological factors and other factors can cause different baseline and affect the comparability before and after the same round. And due to the problem of the sample size, the possibility of making type 2 errors increases. So this is the advantages and disadvantages.
Thank you so much for the summary. It was very complete, actually. And as you're saying, the mainly advantages are patient-directed, right? Now we see that we always see, right, that patients have different response to the same treatment. A treatment that I take for my condition, you have the same condition and you take the same treatment, the results or our experience with that medication can be a little different, and it's because I am a little different from you, right? So when you think about N-of-1 trial, we think about this context of individualized medicine. Can we do individualized medicine? Can we think about a treatment that is specific for each person? But also this comes with costs, right? And the cost is that you have to pay very careful attention to your design, right? That result is for that individual, even though you can have some kind of generalizability. It's not so generous. You cannot have a lot of external validity here. And again, it's the balance between the cost of doing an N-of-1 trial, right, and the benefit of the amount of people that you benefit on that. But for some chronic disease and some for diseases that we still don't have a treatment that is satisfactory for the population, this could be a very interesting design, right? And in this case here, we have fibromyalgia. There is a chronic condition, and that so far we do not have a satisfactory treatment for fibromyalgia, even though we have different pain medications. That would be a very good example in which N-of-1 trial could be beneficial, right? Since each patient is a little bit different and have a little bit different symptoms and have a little bit different way to cope with the symptoms. So this could be a great idea here. Okay, thank you for summarizing the case and giving us the advantages and disadvantages, right? Can someone, I don't know, Keiko, if the same student wants to keep going, but yesterday in the live lecture, some people were asking the difference between N-of-1 trial and a case study. And I noticed some confusion between the students, so I wanted to know if someone can tell us what is the difference between an N-of-1 trial and a case study.
Okay, TM, can you continue? Do you want to continue?
I'm sorry, I'm not very clear with this question, so I'm afraid I can't.
Oh. No problem. Maybe if so, maybe Alberto, you can talk.
Sorry, I'm kind of the same. I didn't really understand the question. Sorry.
Can I try?
Yes, I think
yes,
so.
This is Julia again. I may be wrong, I'll just try. So as far as I know, case study are like not intervention. So you just observe the patient. It can be prospective or retrospective. And in N-of-1 trials, you can interfere with obviously intervention and control. I think that is the difference. But both are related to one subject.
Exactly. That's very good explanation, Julia. Thank you. It's exactly like that. A case study, you don't interfere, you observe the patient, right? It could be prospective or retrospective. And in an N-of-1 trial, you are doing a clinical trial, okay? You have everything else. You have blinding, you have randomization, you do have to assess adherence and everything else. It's a trial, but it's with one person. Okay, so this is a very important thing to have in your mind. A case study, it's not an N-of-1 trial, and an N-of-1 trial is not a case study. Is that clear to everybody? Yeah, I'm gonna move on then. So an N-of-1 trial, again, one patient is involved, it's a single case, but there is a manipulation of the independent variable. So you are manipulating, we are doing an experiment, okay? This only one participant is exposed to all levels of the independent variable. So it's like if it's a within patient design, a crossover design, for example, okay? There are repeated measures and observations. So you measure your outcome several times in an N-of-1 trial. And there is randomization. The only thing here that instead of randomizing the patient to the treatment, you will randomize the way you're gonna give the treatment to the patient. It doesn't make sense. It's a little bit different. We're gonna get better when we think about examples here. So Dr. Sato's idea was doing 2 cycles of drugs, drug A and drug B, right? Within a short period. And he would have cycle A or cycle B and drug A and drug B. But here we would have to remember that that patient have to be randomized to cycle A or cycle B, or changes to cycle A from cycle B, okay? So this would be the randomization in Dr. Sato's idea. We already talked about advantages and disadvantage. So I brought some examples here. I'm gonna try to move a little bit fast, okay? So if I'm going too fast, let me know, type of N-of-1 designs for us to understand how this is different from a single case or a series of cases. So there are different type of N-of-1 designs. One type called alternation designs, in which you change from one treatment to the other, and one treatment is one day, the other treatment is another day, right? So you're alternating from one to the other without having multiple assessments in the same treatment. So here's one example. We have 2 independent treatments, A and B, and we want to do at least 3 measures of A and 3 measures of B. And then we need to have a randomization, meaning all possible combinations of A and B if we have 3 of them. So here you have spelled out all possible combinations. So if I want to have a subject in this, I will have to randomize the subject and see in which one of these possible combinations he would fall in, and I would follow the randomization. Understand? That's what I mean about randomization. You would take all possible of combinations that you could do your trial, and you randomize the patient to that. So in an N-of-1 trial, you should not only come with the idea, oh, I'm gonna do an A-B trial and it's gonna be A-B, A-B. That's not exactly correct, and that's not exactly how it's done. You would have to think about all possible combinations of A and B and randomize your subject to one of those combinations, okay? And this is very important to later on see the significance of our results and calculate if there is effect of the treatment in that result. Another, I think I brought here one example. This is one example of an alternation design. So in this trial, they were checking pain intensity in kids with cancer. And what they were doing here, it was alternation design, meaning one day they would do something and the other day another, following the randomization that they got it. And this was a behavioral intervention. So when they have the ball example, the GPM, these kids could play video game, right? Some online video game. And when we have the square, they would not play video game. They would just stay doing your normal daily activities at the hospital, right? And we can see here that usually when they had the intervention, when they could play video game, we had a little bit less pain. Sorry, I told you the wrong thing. I'm so sorry. So going back, the GPM, it's the control intervention, and the square is the video game. So we can see that when they can play in the video game, most of the times the pain is lower than when they are not playing video games. So this was a video game used as a coping strategy to deal with pain, and they would rate your pain the days they were playing and the days they're not playing, okay? So would be a type of trial used for a drug? Probably no, because drug has carryover effects. Most of drugs have carryover effects, but this type of alternation design is very common in behavior type of studies, okay? And we have also phase designs. We discussed this a little bit yesterday. So it's the most common design. In this design is a sequence of measures that is repeated in treatment phases, right? And several measures in each phase. So here's one example. This is a headache study, and we have here 5 different patients. And what they were randomized here, it was the randomization of changing from one phase to the other phase. So I had here phase A, that it would be control treatment, and phase B, that would be my treatment for headache, right? And what I randomized here is when a patient would come from one phase to the other. So Adam was randomized to 4 points in control baseline, and all the rest of the study he was in the drug, while Rachel was randomized to do this change from phase A to phase B on the day 5, and Cindy on the day 6, Elmer in the day 7, Jane was also in the day 7. See, it was a randomization. I did not choose who would change phase in each time. I randomized the changes in phases, and that patient was randomized to that change. Is that clear, guys? You have any questions here?
Hello, I'm sorry. Is Brigida here?
Hi, Brigida.
Hello. Is this last slide that you show for us, the last one?
Oh, this one?
No, yes, this one. This drug, A and B, BA, okay. And in that period that you are considering between the period 7 and 10, and after between 10 and 13, there are like a lower effect here. And my question here is if this time is like we are considered that maybe there is a different treatment, like or there is no treatment here maybe, and if this time can be considered like the washout period, and this like between the 16 and 19, maybe here we can understand that maybe we have some like a carryover effect here.
Yeah, so even though this was a behavioral intervention, we can see that behavior is harder to measure carryover effect of behavioral intervention, right? But you can see that usually when the kid it's doing the video game, like the pain the next day can have an effect because in the previous day they were so happy, or can have the opposite effect, right? They were happy here on the day 7, for instance, and then on the next day they could not play the video game, the pain went up. But when you look for the 13, we could see that there was a more carryover effect. But it's hard to analyze this carryover effect in this type of the intervention. But how you would analyze this data, you would do the mean of B, because this is an alternation design, this is not a phase design, so the analysis is a little bit different. But we would do the mean score of pain B and the mean score of pain A, and do the difference of this scores, and then we would be able to say if the B intervention was really working here compared to the A intervention. But yeah, looking to the graphic, you could see that in different phases, the kid respond different to the intervention, and there was a different carryover effect of the intervention here. We would have to analyze if, in this case, I don't know, right? But in this case, it could be that some of these days they were feeling more pain, or they were having a new round of chemotherapy and so on.
Okay, so it is a cognitive study. Okay, thank you.
This is a cognitive study.
Okay,
yes, because of this, yeah. But if it was a drug study, you would have to be more concerned about carryover effects. Yes.
Sure, thank you.
Yeah, you're welcome. This one was a drug study in this case, and to be honest, yeah, I don't know by heart now. I'm not gonna say, but it was a phase design in which you randomize the changes of the phase. Okay. So yes.
So in that graph, can you go back? In that graph, does Rachel has carryover effect? That's why she's that pain intensity is decreasing.
So we don't know, right? Because here we randomized phase change from baseline, and baseline here was no treatment, to a self-regulation training. Sorry, he was also a behavior effect. Sorry. meaning like they were doing their normal drugs here on baseline and then start doing a training, like a cognitive training to self-regulate red eggs. But if you see, even if this was drug, we could not say that Rachel has carryover effects here because she's still on treatment phase. So she's still on medication. So it doesn't mean it's carryover effects. Understand?
Okay. And how does carryover effect look like in this graph?
So in this graph, we cannot see exactly, but for instance, let's see if we had, if instead of baseline here, we had a drug A. And the drug A was in fact making pain go away, right? And our patients were getting better with pain. When I changed to phase B or drug B, I did not have an enough washout period. So patients starting phase B already start with lower pain than patients starting phase A, because the drug that they are, the drug that he was on phase A already reduced his pain. So instead of starting with pain of 8, they start now with pain of 4. And they start drug B, but they still 4, 4, 4, 4, 4. And now you don't know if that effect it's because of drug B being able to keep the pain down or reduce the pain, or if it's a carryover effect of drug A because drug A already reduced the pain of that patient.
Got it.
Got it? Yes. That's why when you do crossover designs, we do washout effects. And that's why for N-of-1, if you're testing 2 different drugs, you also need to do washout effects. So besides of randomizing the change of phase, you would have to include a washout period between the phases, right? Here, as we are talking about a behavior intervention, the control or the baseline in this example, it's no intervention. Right? They are not doing the intervention. And when they are randomized to start the intervention. So we don't have to worry about carryover effects because they are not doing anything before. Let's imagine that now we have another phase. So after Adam finishing his treatment here with pain 0, right? We will randomize him again to a phase C. Okay, so we have phase A, that is baseline, phase B, that is the training, and a phase C. Here I would have to be worried about carryover effects because this intervention make my patient have pain of 0. So for me to start phase C and see if phase C or the treatment that I'm gonna test and see works, I would have to wait a little bit, right? Because if this patient starts with pain 0 and keeps with pain 0, mean of C is 0, so C is the best treatment ever. Because we'll have a lower mean than B here, understand? Even though B was the treatment that in fact reduced the pain. Does it make sense? Ahmed?
Yeah.
Yeah?
Yes.
Okay. So we are almost finishing the lecture. So in this example, and it's important to think in a N-of-1 trial that we need to do this randomization, and why we need to do this randomization is to calculate again the probability of having a false positive result, right? Of having a result that is not there, and the randomization will help you with that. And how we do analyze the data for this type of studies. There are several ways to analyze the data. I will show you guys one example here of a package that can help you to analyze the data. There are softwares that help you to analyze the data for and design N-of-1 trials. And how we know if the results are significant. So nowadays there is a whole methodology on N-of-1 trials for us to check if the results are significant or not. Okay, I won't have too much time here, but this is another example. We want to do the phase study. Actually, this is the AB of the previous slide. So let's suppose now we are randomizing the start of treatment B. So we have these 5 different options, right? We could do one measure on A and all the measures on B and so on. So if I randomize a patient here, my patient would have about the same chance to get in any one of these studies. So it's about 100 divided by 5, 20% chance of being randomized to any one of these trials. And we will use this 20% probability of being in one of these trial designs to later on calculate the p-value and see if the results are significant or not, okay? This is exactly what I told before. So we have the measurements, you have our null hypothesis that is the same as trial with more patients. Our null hypothesis, there is no difference between the treatments, and here my hypothesis B, it's better than A, and I have all these probabilities and all these possibilities of randomization, and then I have my data. And when I look to my data, if I don't know which possibilities they are, I don't know what the results are, I don't know if A is better than B, and this is the point, right? The point is you to compare the results that you have from your patients with the amount of possibilities that that patient could have gone on, and that's how you calculate later on the p-value. Okay, I'm just going to move a little bit forward, but I will show you guys a website at the end that can help you to understand, and there are some examples and even Excel spreadsheets that you can download with data for N-of-1 trials and do your own analysis and explore more as you want. So we talked in the beginning about levels of clinical evidence and evidence-based medicine, and we could say that N-of-1 trial is on the top of the pyramid because it's a specific trial for a specific patient, even though we also discuss about its disadvantage too. We do know that this is not a case control study, this is not a case series, it's something that you are actually controlling the intervention and you can change that, okay? And we also know that for some things are not necessarily to do a randomized clinical trial. Should we do a randomized clinical trials to show that parachutes work to prevent death? There is no, go to the literature and see. But everyone that jumps from an airplane uses a parachute. So I know it's a funny example, it's an extreme example, but just to show that, of course, they are on the top of the pyramid, and we tend to believe the evidence of randomized clinical trials or meta-analysis and so on, but these other designs exist because we have more than one type of question, and always go back to your research question. What is your question? What do you want to prove with your trial, okay? And here we can think where or when would we use this type of N-of-1 trial. We already give some examples, for instance, in chronic conditions, in very hard to treat conditions, or in very not common conditions in which individualizing the patient can bring you better results, okay? And then the last thing that I want to show is this website here. Let's see if I'm going to open, okay, here. So we can share. Can you see my internet screen? Hello?
Yes.
Yes, okay. So this is a website from Leuven University. So here they have some R software packages. It's very nice if you have doubts about N-of-1 trial or have interest. They have a package that help you to do single case experiments, calculate your confidence of interval, help you to do the randomization and so on. If you download the zip file, it also comes with some Excel spreadsheets with some data in which you can check for that. So it's one of the packages. There's also a graphic user interface to help you to do graphics and so on. This one is another package that also help you to do single case experiments here, and is online, all available. It's for free. You can go in the website and download and explore. Yes.
Can you share the link?
Yes, I will put
maybe copy and paste in the chat.
Yes, here. Can I see the chat more?
Or later, maybe.
Yeah, I will copy and paste as soon as I don't know why if I share the screen, I can do this. I also added in my slides, so we could. Yeah, it's in the end of the slides. Yes, yes. So it's there. I think it's very interesting, and I think this year it was supposed to be the first N-of-1 conference in Leuven, and it was supposed to be in April, but because of coronavirus, they have to cancel it. But the name was One is Beautiful. It was a very interesting name, yes. So for today, that's it, right on time. Sorry we didn't have more time for questions at the end, but if you guys want to ask something, please go ahead. Hi, Gisela.
Hi.
Hello, is Brigitte here again? Can I ask you a question? I don't know if we can perform like a meta-analysis for N-of-1 trial. I did this question for Professor Fragan, and he said that yeah, we can. But my question is like how we can assess like this heterogeneity for the N-of-1 if the study is not heterogeneous, if it's done in just a person.
Yeah, it's a very good question. We can do a meta-analysis of N-of-1 trial. There is like nowadays a guideline of how to do this meta-analysis and how to assess this heterogeneity, so you can do that. The heterogeneity is also based on the randomization and the probability of that patient being randomized to each one of those different assessments, and you would calculate that based on different possible results. Okay, so it's also a probability, but it's harder to be generous, it's harder to have the same generalizability as you would have with a trial. Yeah, I understand. We don't have a standard deviation, right? But
yeah, okay.
But you can do that. I think in that website, if I'm not wrong, they might have a guideline about how to do the meta-analysis. But
oh, wonderful. I'll check it. Thank you so much, Camila.
You're welcome. Yes. Any more questions?
Hello, Camila.
Hi,
this is Hans. Just a quick question. Initially, I mean, before the class, I understood that when you run interim analysis for safety only, you don't do any calculation, you don't lose any power. But still, I think based on what you have just presented, we need to run some calculations. So that's why even for interim analysis, looking for safety, we need to do the alpha spending, right? Maybe using the pocket method.
Yeah, so I think we discussed this a little bit with Julia, but then when I talk again, it might get a little bit confused. So you would do an alpha spend function for safety if your primary outcome is safety. Understand? So you don't use an alpha spending function and you don't lose p-value if you look for safety when your primary outcome is something else. Like the example of the Serenium trial. They wanted to prove efficacy of the drug compared with the placebo, and they would do an interim analysis for safety. So in the interim analysis, they would not analyze the primary outcome. They would not check the data of insomnia, right? They would just check safety. In that case, you don't use an alpha spending function. But if you are concerned about safety and your primary outcome is safety, right? I have a trial that I want to show that this new chemotherapy method is safe for kids, right? And I want to do an interim analysis of that safety. Now safety is its efficacy. So you would have to use an alpha spending function. And in those type of trials that are not common, okay, then you would have to use an alpha spending function and lose power for that. And for that, they can be a little bit less rigid. You understand? Sorry, I was confused on that.
No, yeah. Now it's clear.
Thank you for asking that. Yes.
Yep. Thank you for your answer.
Yes. Any more questions?
I think everyone is now clear. Yes. I cannot find any more questions from chat.
Okay.
Maybe if we have, we will send you an email.
Yes, this is my email here in the end of the slide.
Yes.
You can send me emails, and I think you're gonna make the slides will be posted in the website, right? So yeah.
Yes.
Yeah.
Thank you for your
Thank you
great
so
lecture.
much. Yes.
Thank you so much. Everyone enjoyed.
Thank you. I enjoy as well.
Thank you everyone for participating, and good luck on your exam. Thank you so much. Bye bye.
Thank you.